So I'm at this place here in Oxford Wolfson Centre for Mathematical Biology. So we're roughly speaking 5 faculty members in mathematical biology, and then about 8 postdocs and about 15, 16 graduate students. And so what I'm going to talk about today, oops, this thing stopped working now. That's strange, it was working before. I'll just use that. Oops. Okay, right, so what I'm going to talk about today is begin by talking about some actually pretty old work, but we've done some bit of new stuff on it, acid-mediated invasion, which links to the title of the conference on sort of ecological aspects, and then talk a little bit about colorectal crypt dynamics. So this whole thing came about as almost 20 years ago now, Gatenby and Galinsky. So Bob Gatenby is a physicist who is now a cancer radiologist, and he heads co-director of the mathematical oncology group in the Moffitt Cancer Research Center, and he viewed cancer very much as an ecological system going on within the body. And so he asked the question, if you consider cancer as a pest and the normal cells as the host, then how can the pest overcome the host? And one of the things that he noticed is that there are many cancer cells phenotype that adopt the glycolytic pathway even when there's enough oxygen for them to use aerobic metabolism. And since the glycolytic pathway and anaerobic metabolism is less efficient for energy, even though you can run through the cycle quicker, the question was, why would an invader use an inefficient energy generating system if it's competing against something? So the byproduct, one of the byproducts of glycolytic pathway is lactic acid, and that is lowers the pH, and that's very toxic to normal cells. So he wondered whether this facilitated the invasion of tumor cells versus the normal cells. So he wrote down a mathematical model. So N1 is normal cell density, N2 are tumor cells, L is lactic acid, everything else is a positive constant. So here we have, if we look at the tumor cells he modeled as growing logistically and diffusing with a nonlinear diffusion. And the tumor cells produce lactic acid. Lactic acid diffuses and degrades, and then the lactic acid kills the normal cells, which are also growing logistically, but are not moving. So that's all realistic. And you see here the idea is that the tumor cells move when the normal cells go below confluence. And then what he found, or what they found, was that there were 2 behaviors to this system. So here now everything's been rescaled to lie between 0 and 1. And so the idea is that you introduce some tumor cells into an initial population, which is all normal cells, and you look at what happens to them. And you get traveling waves. And you get 2 types of traveling waves. You get this type here. So here are the tumor cells, like this. Here are the normal cells. So you see here is coexistence, far behind the wave, far ahead of the wave, everything's normal because the tumor hasn't reached there yet. And then here is the acid profile. And then this is moving in this direction. And then on the other side of a bifurcation parameter, you get this type of behavior. So again, you've got the invading tumor cells, you've got the receding normal cells, you've got the profile, acid profile, so all very similar to here. The difference has been that here you've got competitive exclusion, and here you've got a gap. So this model made a prediction that there should be an acellular gap between the tumor cells and the normal cells. And then he looked at some head and neck cancers, and in about 1/3 of them, he found this gap that was predicted by the by the experiment, by the model. Okay? And then here are also acid profiles. These are pH profiles taken by slicing through the tumour. And the tumour is here, and then the normal cells are here, and the pH values are like this, which means that, of course, the acidic gradient is like that. So that's also consistent qualitatively with what the model predicted. Okay? So then this obviously suggests that what you should do is add bicarbonate to neutralize the acid. And so this they did. So they took some mice, they put tumors into the mice. In one set of mice, the mice were chronically exposed to bicarbonate, so in the drinking water. And in the other set of mice, they had normal drinking water. And then they looked to see how the tumor invaded. And they measured invasion by metastasis. And what you can see here is the untreated metastasis levels are here, with the mean here, and the treated ones are here with a mean a lot lower. Okay, so obviously the treatment doesn't totally stop the invasion occurring, because we would expect that there are many contributions to invasion, and this is just one of them.
Were the mice using all gas often?
Sorry?
Were the mice using all gas often? Those ones treated?
Well, I don't know. Yeah.
No, but this is a very cheap treatment.
Yeah.
Where they count the things, so this will be followed.
Well, I'm coming on to that. Yeah. So there were some problems with this model, which Gatenby and all were aware of. One problem is, if you notice, well, if you look at this long enough, you will see that the normal steady state is not stable. Okay, so you would expect that a system like this, there should be a homeostatic state that's stable. A normal steady state should be stable, because not everybody gets cancer. The normal state of us is a stable N1 equals K, and then there's an invasion. Okay, this doesn't give you that. And that's very easily filled. In fact, this is the model they had. I'm sorry, now I've changed to U, V, and W. So this is N1, N2, and L. And in fact, if you put the competition in here, they had this originally, and then for analysis, they just got rid of the competition terms. Once you put those competition terms in, it becomes a Lotka-Volterra, and therefore you can have a parameter space in which N1 equals a constant is a stable, steady state. The other thing they did was they assumed that the tumor cells were totally resistant to the acid, and they're not. So we put in this thing here, which is a lot smaller than this. So that's the two changes we made. And it turns out, if you look here, that's small. There's a small parameter in this system, which is the tumor cell diffusion coefficient divided by lactic acid diffusion coefficient. So once we've made this extended model, we can do asymptotic analysis. Okay? And I'm not going to go into the asymptotic analysis, because trust me, if I did, you would lose the will to live. Okay? So it's pages and pages of asymptotics. There are many different cases you have to look at, etc., etc. And this was worked on by graduate student Jessica McGillen, who just got her thesis a few days ago, and with Eamon Gaffney. And as was mentioned before, this was sort of done several years ago and appeared in JMB just recently with a long time delay. So this is just to show that the asymptotics, we could use the asymptotics to measure tumor wave speed and to measure the gap. And so here just numerical simulations, and the solid line is the analytic work. Okay? And then what we can do now, you can't see this here, which is a good job, because this is the wave speed in terms of the parameters of the model, but also in terms of various other things that come about from doing all this analysis. But what you can do in the end is you can plot diagrams like this, where you can see how does the wave speed depend on the different parameter values. And you can do the same thing by saying how does the width of the gap depend on all the different parameter values. Because what we'd seen beforehand was a qualitative comparison, a qualitative prediction that was compared qualitatively with data. And so the question is, when we put real numbers into the system, do we get quantitative comparison? And the answer is no. It turns out that if you look at the gap that was predicted and the gap that you see here is a few cell widths. And if you ask the question, what parameters do I need to come up with a gap that is a few cell widths, you find that it's a very, very tiny parameter space that will also give you the correct values for the pH. If you make the assumption, however, that the gap is narrower than what you saw in the experiment, then the parameter space in which you get the gap increases. So what this is suggesting is that really you should see a smaller gap than the gap you actually see. Now, Bob isn't stupid. He'd already realized this. So in fact, and a lot of biologists criticize this because they say this is a fixing artifact. In other words, the biological process that had to be carried out to take this picture, they say, made this gap a lot wider. Okay? Some of them say it made the gap. And then I think it's very strange how the gap happened to be between, this artifact happened to occur between normals and tumors rather than occurring across here or along here or along here. Okay? So Bob, in the paper in the 1990s, actually did the same picture but without using fixing, without using this biological technique. And now you see you get a much narrower gap. So this suggests that this gap actually does exist. It's not an experimental artifact. And it's also consistent with our work that says realistically the gap should be a lot smaller.
But I understand something. In cancer, you have a mixture of cancer cells and normal cells together. So why is this gap? This means that there is separation between them?
For acid-mediated invasion tumors. Yeah, because there are a lot of tumors that invade without using acid. But this is for tumors which use acid-mediated ones. Yeah, that there'll be a gap. In some other tumors, what you find is that they encapsulate themselves. There's actually a protease. They actually encapsulate themselves in terms of protease, and there's a gap between the tumors and the normals. And then there are other ones, as you quite rightly mentioned, that they're mixed up. Yeah.
Is it homogeneous?
Yeah, according to this theory, yeah.
Is it really so? Don't they have mixed normal and...
Well, in this picture here, there aren't actually mixed. If I go back here. I mean, there's a definite boundary between the normal and the tumor. But the thing you've got to think about is that you have to think that this is in part of the tumor. So there may be some other part of tumor over here invading down like this. So that this could be the whole tumor, this could be incorporating the whole tumor. Yeah, so wherever the tumor meets the normal. So not necessarily then that all the tumor is back here. You know, the tumor could be mixed in with the normal cells, okay?
As you made this statement about acid invading,
Yeah.
is this an official classification that some of the tumors are and some are not?
Yeah, so there's some highly acidic tumors that don't invade, and one of the reasons for that is that, of course, there are other things other than cells that the tumor has to invade. And so the tumor has to cooperate with normal cells to produce some of the enzymes that are needed to degrade the the extracellular matrix. And so you can have a situation where the tumor actually isolates itself because it's killed all the normal cells. Because not only is there competition between normal cells and tumor cells, there's a high level of cooperativity between the two of them. Yeah? This would just be extracellular material. So basically everything that isn't a cell. Yeah, yeah. If you think of a tissue, everything in a tissue that's not cells. Okay? Yeah, so this would be collagen, glycosaminoglycans, all the other stuff. Yeah. Yeah. Left there. Now, some of you should be getting very nervous, or even more nervous than you already are about this model, because notice that this gap I mentioned is just a few cell lengths. And therefore, this gap isn't visible in a PDE model. Because when you write down a density, you're making the assumption that you're working on length scales that are much larger than the length scale of a cell. Sorry? About 10 microns, yeah. Well, it's a level of a few cells, and so the question is that when you get down to here, you're making the assumption that you can describe this as a cell density. You're making the assumption that stochasticity and noise is not important here. Yeah. Yeah, so for instance, if there was one cell here, it would be 0 in this picture, but it would totally contradict this because there'd be cells in here. Yeah. Well, we can talk about that. But anyway, what's happened then is that, and I don't have pictures of this, if you actually do an individual cell-based model of this system and put in all of these sort of properties but into individual cells, you still get the gap. And if you then take the statistics of the individual cells and work out the cell density, you get precisely these pictures. Okay? So it does work out okay in the end. Yeah?
So is the gap just dead cells?
Yeah, it would be, yeah, basically it would be whatever these.
So then I don't think there's such an issue.
Yeah, that's right, yeah. I mean, they're basically sort of necrotic cells. Okay? So then Ursula was asking, has this been continued? And the answer is yes. So this was a paper that was published in March of last year, where again they're looking at this picture here. In fact, this is probably the easiest picture to see. And what you have here is, this is where the tumor is growing, and then the tumor isn't growing, so the tumor is red. pH values, here you see low, so that's acidic. Here you see high, so that's alkaline. Okay? So, as has been mentioned before, it's very easy to cure cancer in the mouse, but the question is, can we do anything in the human? And in particular, we know that the human is better at controlling, we are better at controlling our pH than mice are. So what we did then, working with Bob, and in fact Bob Gillies and Ian Roby, who are also, so Bob Gillies is also at the lab with Bob Gatenby, and Ian Roby is in Arizona. What we did was write down a very, very simple model, systemic model of the control of bicarbonate within the body. So this is a whole body model. So what we have here is bicarbonate plus acid gives you CO2. And we have renal filtration that affects the bicarbonate. And ventilation, of course, breathing, etc., that affects the CO2. And we write down an equation like this. So T means in the tumor. So this is B is bicarbonate. So that's the chemical reaction, that chemical reaction here, happening in the tumor. That's that one there. And then this is vascular exchange between the blood and the tumor. So very, very simple model. And then the acid, same chemical reaction, but now we have the tumor production and we have the same vascular exchange. And then the bicarbonate, or the carbon dioxide, I mean, we have chemical reaction, tumor production, and simple vascular exchange. And then for the bicarbonate in the body, so B means body, the same equations as before, except we have ventilation affecting the carbon dioxide. And again, this is just a model that we took from the literature. And kidney reabsorption, which again is a model we took from the literature. So very much the idea here, keeping the thing as simple as possible. It turns out that there are 3 time scales in this, and lots of small and large parameters, so you can do an asymptotic expansion. You can actually find the steady states, get a uniformly valid approximation to the solution of the steady states, and then that means you can do sensitivity analysis. Because remember, what we want to do is we want to see how can we change the pH of the system. So if we've got a formula for the pH steady state in terms of all the parameters, we can ask the question, to which parameter is the system most sensitive? And then we can say, let's change that parameter. So the first thing we did, obviously, was look at the bicarbonate. And so we look at this picture here, and this is what would happen with the equivalent dose. So in other words, you look at your mouse and you see, well, if I give this amount of bicarbonate to the mouse, and then you say, well, we're bigger than a mouse, so let's scale up the amount of bicarbonate and give it to the human, what will happen? So here's the mouse tissue, the mouse tumor, and this is the change in pH that will happen if you give it bicarbonate. So this is a prediction of the model. But this is the prediction for the human. The pH is going to be a lot less. Okay? I mean, the change in pH is going to be a lot less. So this is bad, because this is saying that the bicarbonate will have a smaller effect on the human. But it's consistent with the fact that as humans, we are better at controlling our pH. But another thing that this model brings out, which was something that we weren't aware of, was that in the mouse blood, so in the whole system, there's a huge change in pH. And why that's important is because pH, large changes in pH, if you get a pH that's too high, that's toxic. Get alkaline poisoning. We didn't know that. The way we found that out was when we saw this, we just told the experimentalists just to increase the amount of bicarbonate that's given. And then they said, but then that will kill the person because it's toxic. So now the question is, can we validate this prediction? Well, we can't validate this one because we haven't done any experiments on humans, but this one has been validated on mice. So in this paper here, with then graduate student Natasha Martin and again Eamon Gaffney and their experimental collaborators, were able to validate this result here. And then, because we now have everything analytically and haven't written it down, we can ask the question, can we do combination treatments to lower? So if we found here that by changing the bicarbonate, by administering bicarbonate, we only have this change, we then said, what would happen if you do a number of other changes? And we've come up with a series of predictions here of how you can change various other aspects of the control of pH in the body, and these haven't been tested yet. So these are theoretical predictions. So what recently has been happening is the question, of course, in diet. Our diet will change your pH. And so Bob Bob Gatenby and some of his colleagues have been looking at the effects, like publishing in things like Nutritional Food Science, looking at the buffering capabilities of different foodstuffs. So they have done trials where they've given patients bicarbonate to drink, and they found it very difficult for the people to stay to that regime, because apparently it's very unpleasant to drink the bicarbonate, and people aren't continuing to drink the bicarbonate. So now the question is, can you modify the diet and have the same effect? And so literally within these next few weeks, Ayn Roby and I will be putting in a grant proposal to look at the effects of diet on changes in pH. Okay? So one other thing I want to point out about, and this is something that, in fact, done a lot in the literature in epidemiology and ecology, is done less so in our field, and that is that when we talk about things like diffusion and reaction and everything, we take ideas from chemistry and physics, where the particles we're dealing with are extremely small and they don't have any volume. But of course, cells have volume. And therefore, there's a question about, can you really write down a diffusion equation for the cell? Because the diffusion equation is based on, I'm standing here, there's a 50% chance I can move here, there's a 50% chance I can move there, and that gives you a Gaussian. And if you're a particle, that's probably true. If you're a cell, and there's another cell sitting next to you, then there isn't a 50% chance that you move in this direction, because there's a cell there. There's a 100% chance you move in this direction. So you have to look at the volume exclusion, and that's the sort of thing that Gatenby was trying to get at with the phenomenological d into 1 minus n1 in there. And we've been doing quite a bit of work on how you sort of correct, so to speak, the mean field dynamics by bringing in the fact that you've got that cells have volume. And so one aspect of this is, if you think of cells sitting on sites, and so you ask the question here, what's the probability density that site L is occupied? So that's a one sort of thing there, means that there's an occupation of the cell in site L. And then very easy to write down the equation. How does this change? Well, how it can change is that suppose that there's a neighboring cell, neighboring lattice site that's free, that's got nothing sitting there, and then with a certain probability you move from there to there. Sorry, you move from there to there. I got that the wrong way around. Let's assume that at site L, the site we're looking at, there are no cells. But in the neighborhood, there are cells. Then there's a certain probability the cell will move from there to there. Take away, this site has got a cell in it, and the neighboring site is free, and it moves to there. Okay? Then there could be proliferation, so this would be movement, and then there could be proliferation, and then there could be death. Now, if you make the assumption, mean field assumption of that, you end up getting this equation here, which is the equation we all love to use. That's the Lotka-Volterra equation, competition equation, and death. But this makes the assumption, when you take the limit of this, it's making the assumption that the mean of A times B is the mean of A times the mean of B. It's making that closure, approximation, okay? Which may not be true. So now you see that to close this system, we need to find equations for these correlations here. So this is the dynamics of two neighboring sites. So we started off wanting to work out what is the dynamic of one site, and what we found out is it depends on the dynamics of two sites. So we need to work out the dynamics of two sites. So we work out the dynamics of pairs of sites, They depend on triplets of sites. So it becomes much, much more complicated. And then we have to do moment closure, which as far as I can see is sort of like a bit magical. You just make these assumptions, you have different moment closure rules, and you close the system. And now you might say, well, look, this is ridiculous because I've got the choice between solving this equation or solving this equation here, where you make some moment closure prediction, you use some moment closure, and you've no real reason for making that moment closure, no real justification for it. So why am I going to all that trouble? So here is a competition model, and if we look here, this is like the true solution. So this is taking individuals, having them compete with each other, and you get one individual goes to here, this black, and the other individual is this one, the dotted line. Okay? This is the model that we always use, the Lotka-Volterra model. You see here, it's an error, and this one is an error here. Whereas if you use that horrible system with the moment closure, you almost sit on top of the line. So at this point you could say, but look, the model is making this error here, which is a percentage error, and this error here, which is a percentage error. But how big are those percentages compared to the errors in the biological system in terms of parameter values? And they're probably smaller than the errors from the biological data. So they don't really make any, there's no real, that's not a big deal. Except here. So here is a competition leading to exclusion. And the actual system gives you that the black population A wins and population B dies out. The model with the moment exclusion says population A wins, population B dies out. The model that we all love to use says population B wins and population A dies out. So in other words, it gives you completely the wrong answer. Okay? So this is very depressing, because it means we have to be a bit more careful when we're looking at these types of models. Yep.
How strongly does that depend on the parameters?
Oh, so that's a good point, because, for example, here, these were varying the parameters, and what you can see is here, all the models are really good. Yeah, so it does depend on the parameter values. That's absolutely right.
What about the inversion of the success on an
Oops.
How strongly does that depend on
Well, we haven't looked at how strongly it depends, because here's one where both have got it right, and then here's one where one of them's got it wrong, but we haven't gone in to look at precisely what the parameter values are. Yeah, so that's an important thing. We're not saying that, that the model we always love to use always gets it wrong, because here it got it right. What we're saying is that in some parameter regimes it gets it wrong, and that means we really need to look and understand why that's happening. Okay, so I'm now going to go on to something completely different, which is modeling dynamics in the crypt. So for those of you who don't know what your insides look like, there they are. Nice thing to look at just after lunch. Okay, and so here you have the colon, and here you have the small intestine. And what happens with the colon and the small intestine is that they're covered in things like this, which are called crypts. And so these are indentations into the, so this is what your stomach looks like here, okay? With sort of rice moving along. And then down here, this is into the tissue. And in our small intestine, we have these things called villi, which are jutting out of the tissue. So these villi increase the surface area of the small intestine enormously. And that's important because, of course, it absorbs all the nutrients. Okay? And these villi then have these cells down, these crypts, and these crypts feed the villus. And this is a very bad schematic because there are a number of these crypts that feed each villus. And this is in the small intestine. In the large intestine, there's no villus, just crypts. So typically, crypts have about, I don't know, for a human, about 1,000 cells. Roughly speaking, about 10 stem cells, so to speak. Then a few hundred transit amplifying cells. And then a few hundred fully differentiated cells. So the stem cells divide once every day. The transit amplifying cells divide once every 12 hours. They go through, roughly speaking, 4 divisions before they become fully differentiated. And what's thought is happening here is that as these cells are dividing, the population pressure causes a wave to move up. And so these are being renewed all the time. Okay? And what also happens is there's a gradient of Wnt along the side here, and that's controlling the division, cell division. Okay? And what people like Nick Wright can do, experimentalists now very, very cleverly, is that they can label a cell and then they can track the progeny of the cell as they move up the crypt. So then, if you have an individual cell-based model, so this you could then try to compare with these data. And so that's what we did. So we have an individual-based model where we make, this is a model actually was proposed a long time ago by Maini and colleagues, Marcus Löffler and people, and we just took the model, borrowed it from them. And so the idea here is that you consider cells as being centers, as being point particles, masses, point masses, that are connected by springs. And then when a cell divides, this is just a tessellation for pictorial representation. So when a cell divides, we randomly place the center somewhere along here with a little spring, and then over time the spring natural length grows, and we solve basically Newton's laws of motions with linear springs in order to see how this all reorganizes. And this is in something called CHASTE, which is developed by my numerical analysis colleagues, particularly David Gavigan and Joe Pitt-Francis and Alex Fletcher and James Osborne. And in fact, now CHASE not only does this type of model, it does vertex models and it does POTS models. So basically, it's a framework that does the 3 most well-known individual base models. So you can go into CHASE, you can just choose which model you want to use. You can use it. If you want to compare it with another model, you can use the other model as well. Okay? And so what we can do with CHASTE is that we have a model for Wnt, and we have that Wnt controls cell division in the cells. So the cells have little ODEs going on inside them for cell division, and it affects the mechanics through affecting cell adhesion, and that affects the friction when you write down the overdamped equation for the elasticity. You have a friction term, and that affects the friction term. Now, why do we focus on proliferation and adhesion? It's because in 95% of colorectal cancers, the first mutation is APC, and the APC mutation affects these 2 things, proliferation and adhesion. So we want to see what effects those have. Okay? So here we have 4 crypts, and we play a little game here. So when each cell divides, its daughter cells are going to be the same color. And then when the cells reach the top here, they get sloughed away. And so the game is, which cells win? Is there a winner in this? Okay? Or will they just stay all mixed? Oh, these are the different cells and their children of the same color. So it's basically the cell types. If we go back, it's basically lineage tracing. Yeah, so we started off, if we go back, go forward first of all and then go back, okay? So each cell has a different color, okay? And obviously, so they're different shades of red and blue and green, etc. And so when each cell divides, its daughters will have inherit the same color. So there's as many different cell types here as there are cells. So obviously that's an unrealistic thing, but that's what we're going to start off with. And then we're just following the clones. So what we need here is some sort of music, like game show music going on. And what you can see is a coarsening is starting to happen, because there are not so many colors here now as there were before. And in fact, you can see here, it looks like red is winning out here, this red type of color, and this blue is winning here. Blue is winning here. In fact, it looks like blue is won there as well. So the key thing about this is that after a while, they all become monoclonal. Yeah. They're all included in this. And what you see here is you get monoclonal conversion, which is something that's observed. So it's thought that all the crypts are clonal. They all have one cell type. So even though you have 10 or 12 different stem cells at the bottom, one stem cell's family wins out. Oh, they're all the same. Yeah. Well, so the thing, it's a bit like it's just highly stochastic. It's just that, because the cells are getting shoved out at the top, if one of the cells happens to divide and knock the other one up, and then it disappears, then you've got rid of that cell type, and it's through that sort of dynamics. It's stochastic, yeah. It's absolutely stochastic. Sorry? No, they're all the same type here. Yeah. Yep. No. Everything was the same. Yeah. So in fact, we can see this here. Okay, so because we're mathematicians, of course, what we do is we look at the crypt, which looks like this, and we say, oh, it's a real pity that that's there, so let's get rid of that. So now we have that, and then we can open that, and then now we've got rectangle. So because we like rectangles more than we like test tubes. And so now what we can do, going back to some of the questions you were alluding to, is we can say, right, here are stem cells, okay, represented here. There are far too many of them, but let's just represent them. So they have one property of division, and then these, the transit amplifying cells, have another property of division. And now we're going to color these cells in different color, and here this is a neutral mutation. So it means that these have the same properties as all the other cells. And then we run this. I can get the arrow thing to come up. Yeah, so they just... And so you see they get wiped out. Okay, if we ran this further, they would all get wiped out. Now what we do is we say, let's assume there's an APC mutation amongst these cells. So these cells have a higher proliferative rate and a higher adhesion than all the other cells. And now what will happen to the dynamics? And now you see they actually stay. And if we do this further, so, and this is the stochasticity side of things, we see this one here, and its progeny are all the same color. It looks like it's going to take over the crypt, but that it ends up getting washed out. And here's one where it does take over the crypt. So then we do thousands upon thousands of simulations. And first of all, we do the simulations where there's no mutation. Everything is the same. And we ask the question, what is the time taken for a clone to win? And so we see here that it's like this, typically, 40 or 50 days is where the clone wins. This tells us that every single simulation we did eventually became monoclonal.
All the cells are stem cells, right? And they are competing against each other.
Well, no, so what's happening is that these cells are stem cells, so they divide once every day. And these cells divide once every half a day. So the blue, we're just saying, here's a stem cell and it's starting to divide. What happens to its daughters?
So this blue one is identical.
Yeah, so the only difference between this blue one and all the other cells is that it's blue. That's the only difference. It's the same, yeah. So when we run this simulation, all of these cells are dividing and moving up, but we have just followed one of them as it divides and moves up. But this one is also dividing and moving up, but it's just that color, and then it becomes this color when it goes to here, and likewise with these ones. Okay.
All of this is random, basically, right?
Yep.
So you say the random essentially ended up with
Yeah,
only one,
with one winning out. Yeah,
all descendant of one.
that's right.
Of the blue on the bottom is a descendant of the tiny dog.
Of this one. And this shows the stochasticity, because this one here, it eventually got swept out. But this one, the same simulation, run again, because it's stochastic, it wins. Okay, so what would happen in this case is that one of these other cells would have won. Okay, so we made the wrong bet. If we'd maybe bet on this one, it might have won. Okay, and here we bet on the right one. This was on the one that won. Okay, so basically all of these cells are dividing and moving up. They're all doing exactly the same thing, and due to the stochasticity of the system, we decided to follow this one cell, see how it would do, and it lost, and here it won. Okay. Yeah, so once the cell goes into here, it's doing something different, because these cells are all dividing once a day, and when they get beyond this line here, they divide once every half a day, because there's a Wnt gradient here, and where the Wnt is high, these cells divide less than these cells here. So in other words, if I start off with a cell here, it divides, produces two stem cells. They divide once a day, divide once a day, divide once a day, get up to here, divide twice a day.
They cannot die until they're on the top.
And then at the top, they're just taken away. Yeah.
So in our trip, each trip, actually, all the cells all the stem cells there were created just by one
Yeah,
when we were babies.
that's right. Yeah, so all our crypts are monoclonal. Yeah, yeah. But we don't know which one, because we don't know which one won. Okay.
It varies from one trip to another.
Yeah, yeah, that's right. Yeah. Okay, 2 minutes. Yeah. Okay. Yeah.
Did you ever redo a simulation restarting with the same cell?
Yeah, Those are different cells. But yeah, you can, yeah.
Sometimes they will win, right?
Yeah, that's right, yeah.
And a question on the monochronicity.
Hmm.
If they're all identical, how do you identify them in a real sense?
Well, you can't, I mean, you,
How do you distinguish between nearby crypts?
oh, well, because they're not identical in each crypt is identical, but one crypt isn't identical to another crypt.
But I thought you were always starting off with identical cells in the crypts, just coloring them differently.
Yeah, so
You ended up with one winning and one losing. We can just talk.
yeah, so what this would mean is that, if this crypt was next to this one, then this one would be this type, and then this one would be the blue type.
But can you actually differentiate between one stem cell in one crypt and another stem cell?
Well, you'd have to do it by this labeling. You'd have to do it very early in development. But the way that they've done it in this picture here. See, what they've done here, they labeled one of the cells blue, and then they followed the progeny. And they got this picture here, and that's exactly what we did here. Was we took one progeny and we followed, and that's why we colored them in blue, because experimentally they colored them in blue. Well, no, this is just a normal crypt. Yeah, this is normal. Yeah. So, in fact, if I can just briefly go on, because a lot of the questions you've asked, I can answer them in the next slide. So, one thing we ask is, one thing that we show in this picture here, so just take another 2 minutes, sorry about this, is that what we find is that the original cell that give rise to all these crypts doesn't remain in the crypt, so that there's no immortal stem cell. So in other words, the cell when we were babies that made one of our crypts blue is not sitting there anymore, according to this. It's gone away, but has been replaced by another stem cell. And then another question is, where do you have to be to win? Because this was the sort of questions you were asking, because you were wanting to cheat, because you were saying, well, could I place this somewhere that would give me a better chance to win? And the answer is, yes, you can do that. And that's what this picture is telling us. It's telling us, if you place here, you've more chance of winning than if you place it here, or here, or here, or here. Okay? And nicely, this has been validated independently by a research group just a few months ago in the actual crypt. And then another question you can ask, now when you decide to look at mutants, you can ask the question, what should I do to make sure this cell wins? So one thing you could do is you could say, let's make it more sticky. And this is the stickiness parameter and the chances of winning, the probability of winning. And as you make the mutation more sticky, you have more chance of winning. And then the next question you can ask is that this line here is where, well, there's a line up here, which I haven't drawn in here, where you stop dividing. And now we're going to say, suppose this one cheats and varies that line. Will that help it to win? And the answer is yes and no. So here, if we make that get higher and higher and higher, here's the probability of winning. It first of all goes up, and then it comes down. So then, we'll not go into that, that's an explanation of it. I just want to show this last thing. Now, in biology, people always talk about something called fitness, which I find is a sort of a mysterious thing, because you're fitter if you win, and if you win, you're fitter, the sort of tautology. And so here we ask the question, what does fitness mean in terms of these parameters, in terms of the biological parameters? And what we find is that there is a correlation, direct correlation between fitness and adhesive parameter. and there's no correlation between fitness and proliferation ceiling. So according to this model, if you really want to win and you're only allowed to cheat in one way, the way you cheat is you make your cell stickier. Okay? And then I'll just very briefly mention that I mentioned these different types of models. So one of the other things that we've done is that we've tried to write these models in terms of PDEs. And when we write them in terms of PDEs, we get a non-linear diffusion, and I'm happy to talk about that later. And so what I want to do to end then is just to mention that in some sense these two models, although they're very, very different, they're the same in the sense that they both correspond to an invasion, so to a traveling wave. They both end up having a nonlinear diffusion in them, but they're different nonlinear diffusions. But they're both designed to answer different types of questions. And what I hope is that I'll give you some idea of what sort of questions they can answer. So here are the acknowledgements. So these are all sort of mathematicians. Experimentalists are here, and the colleagues here are the numerical analysts. And then here's the people who funded it. So thank you for your attention, and sorry for running.